Envision this. A crime scene has just occurred. The day after, no eyewitnesses or suspects. Just a few drops of blood left behind. Forensic scientists have to rely on short tandem repeats to analyze the blood left behind. They will have to take the sample and compare them to database to look up the exact individual and lock them up. Simple, right? But what if there is no match? For perspective, the Davis population has 65,000 residents. Trying to find the exact suspect is like finding a Skittle in a bucket of M&Ms. It's just not possible. This calls for forensic DNA phenotyping, the game changer to predict a suspect's appearance straight from their DNA. We'll delve into how DNA phenotyping works, the current works done in this field, and how we inch our way towards real-life genetic Sherlock Holmes. So what short tandem repeats lack in reach? The DNA phenotyping flips the script. Instead of matching the DNA, what if we can take the portion of DNA segments and find out what a person looks like with appearance traits? That'll be pretty helpful. DNA phenotyping does just that with tiny DNA variations called SNPs, or single nucleotide polymorphs. Think of these SNPs as genetic spelling mistakes. Accidentally replacing an A with a G can be the difference between having brown or blue eyes. But how do samples make their way from their genetic makeup to phenotypic traits? We start at the crime scene. Step 1. Extract the DNA from crime scene stuff. This can be like blood, saliva, or other bodily fluids. Step 2. Using the DNA, the genotypes of the SNPs can be extracted with the technologies of the open array system, a system that identifies the variations of these SNPs at hundreds at a time. After getting the data out of these variations, it will be fed into prediction models like IrisPlex for eye color or BLR for freckles, predicting the actual phenotypic traits to a certain accuracy. This can help generate a quote-unquote profile to the basis of identification. These steps are a strong basis of how research is done in this field. While we can speculate what these SNPs may predict, at the end of the day, population does matter in the world of genetics. A certain SNP for lighter hair in Norwegian populations may stray too far away from, say, the population of East Asians. This is why the custom SNP panels created for the Korean population identification by Seri Lim's lab is crucial to having a well-rounded ancestral component that further improves accuracy. Further upon the progression in this field, the bread and butter of forensic DNA phenotyping confidence right now lies within pigmentation. The prediction in eye, skin, and hair pigments are around 80% accurate, accredited to the well-studied OCA2 and MC1R genes. Pigmentation is also genetically easier to work with as opposed to new trickier traits like freckles or hair types, seen with projects done by Hernando and Prospectus Lab respectively. With fewer SNP correlation tests and important environmental variation like sun exposure, it shows common challenges and room for improvement when dealing with more complex traits. For this class, to relate back to healthcare, here's a twist. The same technology used to identify different pigments and phenotypic traits could also be applied to the rates of melanoma risk, a type of skin cancer caused by pigment-producing cells. Those with a certain MC1R gene variation that causes red hair and freckles can also increase the risk up to 40% chances to develop melanoma, even without the environmental factors that could attribute to developing this disease. Knowing this can help providers identify high-risk patients early and provide personal preventive care to reduce such risk before it gets worse. This is one of the many ways that the development of genetic knowledge can have on the health field. So, forensic DNA phenotyping, from SNPs to identifying traits of suspects. This will revolutionize forensic science and genetic knowledge, one genetic clue at a time. Stay curious.